Hello everybody, and thank you for giving me an opportunity to present my work here in IIT Jammu. And thanks to the organizer, Vishwanath Chakraborty and others. The title of my talk is Emergence of Structure and Organization in Bio-polymeric Systems, a physics perspective. So, what I work on is the broad area of soft matter, which is an application of statistical physics to look at structure formation and the dynamics of macromolecular systems at the length scale of micron to nanometers. And this soft matter systems in the last decade or so has also been used to understand physical phenomena at the inside the cell, because the cell has a length scale of 10 microns or lower, and the proteins and the molecules and all the structures that are formed, the organization that happens and the dynamics that happens at the length scale of, again, microns to nanometers. So soft matter physics with appropriate equilibrium or non-equilibrium dynamics incorporated is the right thing, or is the appropriate physics approach to look at the dynamics within the cell. So that's why physicists are getting to use soft matter to look at also biosystems. There are some people looking at tissues also. So that's how the word bio comes in, but I'm essentially a physicist. And today I'll be talking to you about two different topics. So the 25 minutes or 30 minutes I'll be spending on talking about bacterial chromosome organization. Okay, and where the basic message is a certain crosslinks or constraints at certain part of the bacterial chromosome, which I model as a polymer physics. So certain crosslinks, confinement, and crowders played the pivotal role in bacterial chromosome organization. So that's the thesis, and what these terms mean we shall explain as I go along in the talk. And furthermore, in the last part, I'll be also talking about spontaneous helix formation in semi-flexible polymers. So helixes are ubiquitous in nature, and all this work has been published. The bacterial chromosome work has been published in 18 and 19 in Bureau of Physics Letters and Journal of Physics Condensed Matter. Also, the part 1 and part 2 back-to-back papers in Journal of Chemical Physics, as well as and this work has been published in Journal of Physics Condensed Matter in 2020. So I shall begin with this bacterial chromosome organization, and that work was done with Tejal Agarwal, a PhD student who has graduated out, and my collaborators have been Farhat Habib and GP Manjunath, who are more biological. So GP Manjunath is a biologist, and those are the people who introduced me to the problem. The experimental data that I shall use to model or as an input to my theoretical calculations, computational calculations, were taken from these two papers, and this was processed by these two guys, Farhat and Manjunath, and then given to me, which I used as input to my simulations. Okay, with this background, let me introduce what is the DNA. Because there's a physics audience, I think it would make sense to go through the basics of how DNA is organized inside the cell. Just to give you an idea of length scales, the human DNA has around 6.46 million base pairs, and so there are 46 molecules inside each cell. And if you take all of these molecules and stretch them out and put end to end, then it would cover around 2 meters. So 2 meters of this DNA chromosomes molecules are basically packed inside a nucleus of 10 micron cube. And here, this is the familiar DNA helix structure, which you might all know about, and that is 2 nanometers in diameter, and each base pair is 3.3 angstrom, so that in a one helix pitch, there are around 10.4 base pairs on an average. Now, this DNA molecule, this is the basic DNA double helix structure that is not typically lying inside the nucleus in a random fashion. Instead, it is wrapped around certain proteins called histones to form the so-called 11 nanometer fiber, and that in turn gets assembled or aggregated into the so-called 30 nanometer structure. So 30 nanometer fiber. So now the organization from the 2 nanometer fiber to the 30 nanometer fiber is somewhat understood. There are of course a lot of open questions, but how this 30 nanometer fiber gets organized to the at the micron length scale, or is there any organization at all or not, is a question of open is an open question, is a matter of debate, and that's the length scale which we will be studying. Except that I won't be studying for higher organisms like humans, which have a plethora of proteins doing a large part of the compaction. Furthermore, these are a large number of polymers, DNA polymers, which are present inside the nucleus. I'll be looking at simpler systems called the bacteria, because the bacterial systems I'm choosing has just one single DNA. They have fewer number of complications in terms of proteins. So I'll be looking at the E. coli bacteria and some other bacteria as well called the Colobacter crescentus. But again, to give you an idea of the length scales involved, so this is basically an E. coli cell, and its DNA has been made to burst out of the cell by using by basically making this cell wall softer. And these lines that you see around are the DNA, and this is the basically DNA cell. You see, it's extremely long. The basic message is the amount of DNA contained or packed inside the E. coli cell is about 1,000 times its cell size. The E. coli DNA, if it is completely stretched end to end, is around 1.1 millimeter size, and it is packed inside a cell which is around 1 micron in width, around 5 microns in length. And this picture has been taken from here. So the broad question is how or what is the mechanism is this DNA packed inside the cell? By the way, bacteria does not have the nucleus, right? So it's all over inside the cell. The question is whether it is packed as a random polymer or not. And we already have experimental evidence that DNA is packed inside the cell not as a random polymer. So these are experimental data obtained using biochemistry methods called the chromosomal contact maps. And what you do is plot both on the x-axis and the y-axis the segment index. What is the segment index? So suppose you have a long polymer chain, and the first 1,000 base pairs, you call it segment number 1. The base pairs from 1,001 to 2,000, you call it segment number 2, and so on and so forth. The E. coli bacteria, or sorry, the secrecentus bacteria has around 4 million base pairs. So you will have 4,000 segments, each containing 1,000 base pairs. And so you are plotting segment index on the x-axis as well as on the y-axis. And what these colored patches show is when you have more color, it shows that it has a higher probability of finding two segments close to each other in physical space in XYZ coordinates. Okay, not along the contour, but XYZ coordinates. And so this is data from the bacteria of C. crescentus. You see that there are certain square patches. Moreover, there's a dial here. And this is data from the human chromosome. And again, you see certain squarish patches all over. Right, and this is data taken from this paper, Lieberman and Eden, for the human chromosome 14. What is the basic message? The basic message from this slide, which I want you to take home, is the DNA is not organized randomly in space. It's not a random coil polymer inside the nucleus, as the case may be for humans, or inside the cell for bacteria. And there are certain higher probabilities to find certain segments closer to certain other specific segments. Okay, So because if it was a random coil, the probability distribution this column up would look very different, right? The probability goes as 1 by r as you go farther along the contour, but this is a very different. This has different square patches, and the broad question we try to address is take the bacteria C. crescentus and take the bacteria of E. coli and take such information from color maps and figure out how they get arranged. So what my experimental colleagues did in 2016 is walk into my room and said, if I give you data like this, chromosomal contact maps like the one I showed here, taking this data, can you predict the 3D organization of the DNA, which can later be experimentally verified? So I'm not the only person who's working on this problem. There are folks from MIT, and there's a UK group, there's a European group, including Debashish, who just gave the talk before me, who are working on such class of problems. So what we observed, or what our primary observation is, or this is the thesis that we started with also, and later by calculations proved it, that crosslinks, so you have a long polymer chain and you cross-link certain points, or you put in constraints between certain segments of the chain. So cross-links at very specific points along the chain leads to the organization of the DNA polymer. And of course, you want to put a very minimal number of cross-links, because if you put in constraints, of course, the number of degrees of freedom decrease. So you really want to put in a very small number of constraints, and the cross-links are not implausible, because in the biological system you do have DNA binding proteins which hold certain segments of the DNA polymer together. So the question is, which segments of the polymer should we cross-link? So this data we took from the experimental contact map. So we took so from the contact map, it gives you the probability that certain segments are found close to each other. So we took the position of the segments which have the highest probabilities and cross-linked them together by permanent cross-link. And we saw, or we checked out whether this organizes the rest of the chain. And of course, again, as I told you, we need to use a minimal number of cross-links. So rather than use the position of crosslinks from the biological data or the experimental data, the contact map, we also had equal number of crosslinks or constraints, but put at random position along the chain. So the number of constraints are the same, but we are not putting them at very specific biological positions. And in that case, the nature of organization is different. It's more diffuse. And we tried this out with 10 different random CL sets, crosslink sets, the position, the way you crosslink, and it always gives a very diffuse organization. So before going on to the calculations, I want you to take this message. So E. coli, which has 4.6 million base pairs, we modeled it by 4642 monomers along a ring polymer, so that each monomer is representing 1000 base pairs. And we saw that if we just do a Monte Carlo simulation without any boundaries, then 82 crosslinks, if you have just 82 crosslinks out of 4642 at very specific positions along the contour, that leads to organization. And if you put this DNA polymer chain in a cylindrical confinement, because after all, in the in nature, this DNA polymer is confined by a cylinder-shaped, approximately cylinder-shaped E. coli cell wall. So then you need only 38 crosslinks in confinement to get some organization of the DNA polymer, which we again, of course, match with experiments. For C. crescentus, which has only 4 million base pairs, which we model as 4017 monomers. Again, one monomer represents 1000 base pairs. You need 60 crosslinks without confinement to get organization, and 33 cells if you use a cylindrical confinement. Okay, with this I shall go into the details of the calculation, but this is the basic 10 take home message, that if you cross link very specific positions, then you get an organization of the bacterial DNA polymer. Okay, so details of the model. I use an extremely coarse-grained bead spring model of the polymer. I already told you that E. coli we model by 4642 monomers, and C. crescentus by 4017 monomers, so that each coarse-grained monomer in our model represents 1000 base pairs. The persistence length of DNA is 50 nanometers or 1000 or 150 base pairs. So when we are modeling one coarse grain monomer by 1000 base pairs, we are well above the persistence length. So it is completely a valid assumption to take one bond, to first of all take the DNA polymer to be a flexible polymer. We are not considering semi-flexibility. And furthermore, we can estimate that the length scale of the each bond in our simulation is around 100 to 150 nanometers, and we are looking at organization at the micron length scale. The DNA polymer is a ring polymer. We allow excluded volume interaction, but the excluded volume interaction is extremely small. It is just the radius is only zero point 0.2 times the bonds between the beads, which we model by suitably truncated Lennard-Jones interactions. As a consequence, chains can cross. Now, you might find this to be weird because in the standard polymer physics, polymer chains cannot cross. But in the biological system, there are proteins which actually cut the DNA and allow chains to cross. So this is an extremely important and also valid assumption that we use, and this really helps to organize our polymers, this degree of freedom of allowing chains to cross. The crosslink between monomers, certain monomers, AB pair of monomers, is modeled by the spring interaction again, where the length of the spring is the same that we use for the rest of the chain. Now, what we know from traditional polymer physics is that if you put a polymer in solution, then due to thermal fluctuations, it can take all possible conformations. It remains almost like a random coil. And the first question to ask, that if we say that a polymer is getting organized, how do we measure organization where we know that the polymer is going to explore different conformations? Okay, so what's the measure of organization? So suppose this is a ring polymer, and we want to first address the issue of how do we measure organization. So suppose there's a ring polymer and 50 monomers along the chain is considered as one segment. So 50, remember that each monomer was representing 1000 base pairs. So 50 monomers is representing 50,000 base pairs, 50 kilo base pairs. And what we are going to look as basically calculate positional correlations between different segments. Is it that at the large scale, at the micron scale, certain segments are preferentially found closer to each other? Now, this is what we started out from the experimental data, but we are taking a very small amount of the experimental data, putting in into our simulation the position of crosslinks, and we are asking whether the entire bigger polymer picture at the micron scale is getting organized um, uh, at a much larger scale. So we are using an extremely small amount of experimental input, putting it and getting out positional correlations as well. Okay, or if there's no position correlations, we will see that in our data. So this is a quantity, something like a pair correlation function, but now here looking at the distance between each pair of segments. The other thing that we look at is also angular correlation. So that's data which I'm not presenting because it will take some time to explain that. And a simpler thing to look at is whether certain segments are always found towards the inner region of the DNA polymer coil, whether certain segments are always found on the periphery, on the outer side of the DNA polymer coil, and so on and so forth. If starting from very different initial conditions, because of the presence of crosslinks, they always find some segments always at the center and some particular segments always at the outer, then we would say, well, these crosslinks are helping the polymer to organize themselves, right? So these are the two data I am presenting, but of course in our paper we had to present much more data to establish our claim of our thesis. Here I'm presenting only two. So here is data, the probability that certain segments, the x-axis is a segment index going from 0 to 80, because there are 80, we have divided up the entire ring polymer into 80 segments, and this is data for E. coli. And the question we are asking is whether certain segments, is around 30, are these found in the inner region or in the middle region or the peripheral region. So here we see that certain segments, say around 40, there is no probability of finding them in the inner region. Some other segments, say suppose 10, they are preferentially found in the inner region. There's a very high probability of finding them into the inner region. These error bars come from 9 independent runs. So basically in all the 9 independent runs, segment numbers say 10 and certain other segments are always found in the inner region. Some are never found in the inner region, and certain are found in the inner region but with probability, well, less than 1, like zero point 0.5, because sometimes they can move out into the middle region. And there is a similar data set for the outer region, where you say that okay, certain segments are always found on the outer region. So this is already like preliminary data which shows that seems to be some organization in the DNA polymer due to the presence of crosslinks. And basically we have used 82 effective crosslinks. This is data without confinement. And the next thing we look at is positional correlations, where just like in experiments, both the x-axis and the y-axis is segment index. It goes from 0 to 80. The experiments, it went from 0 to 4000. And just remind you, but here it's going from 0 to 80 because we have further co-screened the data and put in crosslinks at only very specific 82 positions for the E. coli data. And here we again see squarish patterns, blocks of probabilities which are blocked together. Such blocks are formed, and certain segments are preferentially found near certain other segments. So there are positional correlations, strong positional correlations between certain segments of the gene. Okay, so this is data from one run, but in this plot we show data from 3 different runs, and we have 9 independent runs, but I'm showing only data for 3 independent runs, starting from very different initial conditions. These are Monte Carlo simulations, and what you see is across these 3 look very similar, which means starting out from very different initial conditions, they statistically, not exactly, it's not like a solid, it's not like you are organized like a solid where each particle is sitting at lattice points. Here the polymer chains are much more floppy, but within that, statistically, there are strong positional correlations between certain segments of the chain, and different runs get organized in the same manner, starting out from very different initial conditions. And all that I've put in are these very specific position of the crosslinks. This is data with an equal number of crosslinks, but very randomly chosen, where the position is randomly chosen. You see that qualitatively, even by your eye, and we have done quantitative calculations as well, that the nature of this data and this data is very different. Here the correlations are all very diffuse. Every polymer segment seems that it can be closer to any other polymer segment. It's more like what I would call a bit of a random organization, where this is organization nice in terms of blocks where only certain segments can be closer to certain other segments, right? So we have compared with random crosslink case as well, equal number of crosslinks. And finally, taking this data, we decided to compare it with experiments. So we coarse-grained the experimental data into 80 segments, and we plotted it only the highest probability. And this is experimental data coarse-grained for C. crescentus. This is experimental data for E. coli, and this is our simulation data. Now, it will take you some time to actually set point by point and compare, but what I can tell you that the E. coli data, the simulation data with a very small number of constraints matches rather well with the experimental data. But the crescentus data, in the experimental data, you have this other, what is it called, diagonal, which we don't find. So something is not right in our model. We have to add something. There's some other phenomena at work. So what could be this phenomena? We put in confinement, we put in a cylindrical confinement because the E. coli cell is basically a cylinder. Well, at the end there is some there is a hemisphere, but that we have neglected. Okay, so the aspect ratio for the E. coli cylinder, E. coli cell wall is 1:5. We started with the 1:1.75, the aspect ratio of the cylinder. And what we did is take a polymer without crosslinks and let it equilibrate within the entire big cylinder. And this is the distribution of monomers along the length. This basically monomers are distributed all over the cylinder, much larger cylinder actually, with the aspect ratio of 1.75. And after that we put in the crosslinks, and then it basically, because the presence of the constraints, the extra constraints that we put in, and we put in a much smaller number of crosslinks, so 26 and 33, we tried it out with different number of crosslinks, and the polymer basically shrinks and occupies area, length only 1:5. So basically this part is left empty. We started with a bigger cylinder, but the presence of crosslinks, it shrinks and occupies the space at the ratio of 1:5, what is seen in an experimental system. For Colobacter, we know that one end of a particular segment is attached to uh, one end of the cylinder, and that's exactly what we did as well. Okay, uh, and whereas for E. coli, there is no such constraint. We just put in the crosslinks at the appropriate positions, and we again plotted the positional correlations in the absence of crowders and in the presence of crowders. So now, what are crowders? In the cell, other than the DNA, there are many other organelles, many other molecules, ribosome, mitochondria, many other things. And as the consequence of the presence of these other molecules, they're called crowders, is basically it compactifies the DNA. At least that's what some of the preliminary research shows by other people. And we have modeled the effect of these crowders, this compaction, by the presence of other molecules, by a very weak effective attraction between monomers. And this is data with crowder incorporated. Now, when we do this, we basically compare the data for Colobacter. This is experimental data, this is simulation data, and we have taken the points with the highest probabilities so that it's easy for the eye to look at. And then we see that the data which previously, in the absence of confinement, didn't match, theory and experiment, our prediction didn't match, that is matching. Just to remind you, we are starting from the experimental data. We are using very few of the experimental data to do our modeling. And then once we get the entire organization, we are comparing it back with the experimental data. E. coli was already matching without confinement. It matches even with confinement. And Colobacter data, that simulations and experiment was not matching, but now with the effect of confinement, it does match. Okay, so with this, I come to the conclusions of the first part of my talk, where we want to say that we also observed that the position of the crosslinks seem to be clustered in physical space for when we use biological data, whereas for random position of crosslinks along the contour, they are spread all over. Now, whether this has any some physical significance or not, that we are basically, it's under investigation still. Okay, and the basic message is what I already said, that around 33 or 38 crosslinks plus confinement plus crowders organize DNA at the micron length scale, which we have carried out for two bacteria, because one could be luck or serendipity, but with two secrecentus and E. coli, we think that crosslinks do play a role in the organization of bacterial DNA at the micron length scale. And I already mentioned that these are all published data of 2018 and 19. Now let me come to the second part of the talk, where I shall talk about spontaneous helix formation in semi-flexible polymers. And this work was carried out by Debarshi Mitra, uh, there were, I want to also acknowledge very useful discussions with K. Guruswamy, who was in NCL before and has moved to IIT Bombay now. He's a collaborator. So what is the broad question? The broad question we are asking is, can a helical conformation self-emerge in an elastic polymeric filament? The elastic polymeric filament, I'm saying it's where the length scale is an order of nanometers to microns, so thermal fluctuations are important. Can it self-emerge where there are no torsional potential? See, if you put in torsional potential between the monomers or in between the elastic filaments, yes, you are binding them, you're forcing them to form helices. But I want to look at helical conformations to self-emerge without the use of torsional potentials or hydrogen bond-like interactions, which basically stabilize helices in the DNA or say in the protein. Okay, so can it self-emerge using much more generic potentials? And of course, helices are ubiquitous in nature, not only in DNA and proteins, but helices are also being used for microrheology and springs in NEMS and MEMS systems, and so on and so forth. So if we can figure out a way how to get stable helices, it would be wonderful to design also future springs at extremely small micro scales. The motivation of the problem came from the following: by some experiments by K. Guruswamy and Bipul Bishash, who took micron-sized colloids. Colloids are Brownian particles. In case there are any students, so I'm just clarifying, these are spherical colloids on which some polymer has been adsorbed. So these series of colloidal Brownian particles were with adsorbed polymers were put along a chain. They were aligned along a chain by applying some external electric field, and then these polymers on the surface were cross-linked. Green dots that you are seeing, they are cross-linked. So now once you remove the externally applied electric field, these crosslinks hold the colloids into place and they become a rod-like polymeric chain. Okay, so you have made is essentially a polymeric chain made up of micron-sized colloids, as a consequence of which now you can look at the polymeric dynamics with the naked eye, because your constituent monomers are not atomistic sized particles but micron sized particles, and you can use visual optical methods to study those. And this is a picture of such a chain, except that what they did is put this polymeric chain made of colloids in a bad solvent. So basically the solvent wants to sorry, the polymers on the surface wants to avoid the solvent, and it basically took up a helical conformation. And the question is how and why. So this was the starting point of the problem. We have solved this particular problem also, and the work it is being written up with K. Guruswamy and Bipul Bishash. But the problem I am talking about started from this broader question, but we did some other stuff, right? So we are not going to explain this, but this was just the starting motivation. This experimental data was the starting motivation to figure out how to find a helical organization, self-emergent helical organization without chemical specific details. Okay, so this was the using very generic potentials. So have we done it? Yes, we have. So this is basically a bead spring representation of a polymeric chain, which is perfectly straight initial condition. And when then there are thermal fluctuations, they basically become a bit like this. And what we have also added is unscreened 1 by r Coulomb interaction or 1 by r cube interaction, two different cases, two different studies, and the consequence of which they become helical. It's more of a helical instability. So if you have a simple polymer chain and you introduce, suppose you make it suddenly charged by 1 by r, so there's repulsive interaction, they become helical, but these helical helices are not stable, they basically dissolve away in time. And I think seeing is believing, so I'll show you a quick movie. You'll see that there's an initial straight chain, and because of the 1 by r interaction, it gets some helical order. There's some repulsion, and that develops some helical order. Now you might and you should, so you see that there are these turns which are representative of helical order. Now you could find this strange, how can 1 by r interaction with a spherical symmetric interaction produce a helix, right? We have not put any torsion potential, we have not put any bend. It's a semi-flexible polymer, so there's an energy cost for bend. So this is which I'm going to quickly try to show that it does form, though it's not a stable one, and give you the reason. The way we have modeled the system is use again a bead spring model, harmonic springs connecting adjacent beads. There's a bending energy cost, epsilon b cos theta, so that if a triplet of monomers is perfectly straight, the angle between them, this theta angle between bonds is 180, then the energy is minimized. Any bend away from 180 has an energy cost. In addition, there are two cases. We put a 1 by r interaction, repulsive, or a long range 1 by r cube interaction. And in both cases, we see helical conformation on introduction of this repulsion. And no counter ion, so I use 1 by r, not screen Coulomb, which might be a bit difficult to realize experimentally, but not impossible. And I'd use Brownian dynamic simulations for our study. This is the relaxation time. This is the Brownian dynamics relaxation time. So time will be measured in units of the diffusive relaxation time. So there are two order parameters that I use, H2 and H4, to identify helices. We will be particularly using H2 because for the H4, suppose part of the chain forms a clockwise helix and part of the chain is an anti-clockwise helix, then H4 will give 0, but H2 will still show helicity. So basically when you H2 and H4 is 1, you have a perfect helix, and when it is 0, you just have a random polymer coil. Okay, so random polymer coil, and even if it's semi-flexible, a bent coil. So if this has finite non-zero values, it shows the presence of helicity, and how it is defined I shall not say for the sake of time. And what we do here is take a very rigid polymer chain. It has 60 monomers in the chain, and its persistence length is a measure of its rigidity. and a high persistence length means it's very rigid. A low persistence length means it is relatively flexible. So here we take the persistence length to be 6 times its contour length, or more than 6 times its contour length. We start with a bent configuration, but then allow the polymer to relax, and it becomes relaxed. It becomes a semi-flexible polymer. Fluctuating end-to-end distance fluctuates due to thermal fluctuations. Here, that's what we see. And there are 60 monomers. 60 E is the length. And if we introduce 1 by r repulsive Coulomb interaction at time, suppose 330 or 333, then immediately we see a spike in the helicity. H2 becomes non-zero. Previously it was fluctuating around 0, but which says that it picks up a helix or helical order, and then it again gradually dissolves. The helix dissolves and becomes 0 again. Okay. Now the question is, well, there should be one more. Yeah. So now if we take another stiff chain with 60 monomers, the persistence length is half the length of the polymer chain. Okay, then if you switch on the Coulomb interaction, we don't see any helicity. It cannot form helices. But suppose we stretch this relatively stiff polymer by applying a stretching force of 20 kbt by A, so that the end-to-end distance again fluctuates around 60. So it's basically straight, but now it is fluctuating because of thermal fluctuations. But there's a force applied. You have stretched it, and then switch on this repulsive interaction, Coulomb interaction. Again, it picks up a helicity. It slowly decays to 0. So there is something, and this is repeated also for 1 by r cube interaction. Okay. So what I want to say is, KBT, the fluctuation seems to play a role, because at KBT, when you start with a straight line configuration, no helices are formed as expected. But when you start with even at KBT with some positional fluctuations, not a perfect straight line, you get some helices. And I will briefly tell you how and why helices are formed. So suppose there's a perfectly straight polymer chain at time t equal to 0. Thermal fluctuations will put in some conformation, some bends in the polymeric chain if it's a semi-flexible polymer. Now, when you suddenly switch on repulsive Coulomb interactions, then all of these monomers will try to move away from each other using forces parallel to the bond vector. As a consequence, very sharp kinks are formed. So basically, when you switch on Coulomb interactions, you have a polymer chain with extremely sharp kinks, very high bending energy, and this bending energy the polymer tries to lose or minimize thereafter by forming a helix. This is the basic mechanism. This mechanism has been explored in greater detail in our paper, where we have followed the spring energy, the bend energy, the repulsion energy due to Coulomb or 1 by r cube interaction, two different cases. It's a function of time, and have established this mechanism. Furthermore, as I said, that these when you switch on Coulomb interaction, repulsive interaction, strong repulsive interaction, these chains form helices, but then they dissolve in time. But suppose you apply a Coulomb potential as a function periodically. Suppose you have put in a periodic potential, 1 by r potential, which increases with time and decreases in time, right? Cos square 2 pi by with a certain time period. Then you see that the H2 basically, so every time this Coulomb potential is switched on, it forms a helix, then quickly dissolves, then forms a helix, then quickly dissolves. And what I have plotted is H2 as a function of time, and here I've plotted a zoom plot. So basically, every time you switch it on, the H2 increases in phase with the external perturbation of switching on a 1 by r interaction. So you have a continuous helix formation and dissolution, and we have very similar data with 1 by r cube repulsive potential. So what I want to finally say is, see, both the semi-flexibility of the finite bending energy is important for the formation of helices. So is strong Coulomb repulsion, or 1 by r cube repulsion. So the strength of that repulsion is basically denoted by epsilon T and epsilon C. So we have plotted a phase diagram, or rather a state diagram, of when we get helices. So a yellow color H2 means we get helices, whereas if you have extremely stiff polymers, then basically helices are not formed. Right, so because sharp kings are not formed. But if you ramp up the values of the Coulomb energy, then you start getting helices even for higher values of the bending energies. But of course, if you go to very high bending energies, so that a polymer wants to be absolutely straight, LPs of 2000, say, then again you don't get helices. I have got We have plotted a similar phase diagram, but when state diagram, when you get helices and when you don't. Basically with this 1 by r cube potential, you have longer lived helices, and also you get to see helices for a larger. You have the state diagram. Just to tell you the value of the Coulomb interaction repulsion that we had to use. They are extremely realizable in nature. I didn't have to put in weird values, but they are unscreened. So the charges that we worked with are in the range of 12 E. On a length scale of 10 nanometers, a DNA chain is more charged than this. Okay, so the basic message is the right balance of spring, bending energy, and repulsive energy produces uh, helices, but which dissolve away in time. I think I have already taken up my time. These details are present in this paper. Journal of Physics is also present in my website, and I thank you for your attention. And I would love to take any questions if you have any. Thanks.